Hi everyone. In this video, we're going to see how glycogen metabolism is regulated. This is one topic that really contributes to a huge fraction of chronic diseases in the US population and in the world, really. What we're going to be talking about is essentially diabetes, the metabolic consequences of glycogen reserve mobilization by insulin. And so we're going to see how that is regulated. And as I said, the consequences couldn't be more devastating and more of a public health concern than a lot of the other things we have talked about. All right, so let's see how we can break this down and make it easier. I have to say that when I was taking biochemistry many years ago as an undergrad, this part really confused me. So we're going to be talking a lot about post-translational modification of enzymes, of proteins, by phosphorylation, and the outcomes of those modifications. So bear with me. And some of the names, they are really inopportune when we're talking about phosphorylations and phosphorylase, which is the enzyme that breaks glycogen. Things can get a little confusing. So let's take it one step at a time. First, you need to remember that one way you can get activation is if you have a double negative, kind of like a double negative in a sentence. It gives you an affirmative. So this mode of removing an inhibitor is very common in biochemical control systems. So keep that in mind. I'll try to keep the color coding the same. So every time you see red, that means inhibition. Every time you see green, that means activation. All right, so here we have glucose and how we put into glycogen and how we mobilize our reserves glycogen to generate glucose. This is the stuff we have already discussed and seen in the previous videos in proteins, in glycogen synthesis and glycogen degradation. So the thing you need to remember here is that insulin wants to put it away, wants to put glucose away into glycogen. So you eat a meal that's heavy with sugar. Insulin will tell the cells to take it and put it away in glycogen. So insulin will stimulate glycogen synthesis. And at the same time, doesn't just stimulate glycogen synthesis, it really actively inhibits glycogen degradation, okay? So what you expect to see from in the next slide is that insulin will pretty much affect the enzymes of glycogen synthesis and degradation, but in opposing ways. Okay, the outcome will be different. Will be different in each of the two cases. So let's see how that happens. So modification of proteins by phosphorylation is a very common and ancient way to regulate their activity. How common? Well, this is something that has been studied a lot in the last few years. Methods became available. So the human genome has about 500 different protein kinases. These are enzymes that modify other proteins, and they put a phosphoryl group on them. So here, for example, for glycogen synthase. So it's very common. How many proteins are modified? About a third of the human proteome actually is modified in this way by phosphorylation. So it's fair to say that life as we know it depends on phosphorylation. What happens now when you phosphorylate the target protein? It depends. In some cases, nothing happens. In other cases, the one you see here, okay, the outcome of the phosphorylation is inhibitory. So it converts. So you take an active enzyme, glycogen synthase, okay, which we have seen what it does. It sticks, one glucose after the next and makes chains in glycogen or glucose molecules. But if you phosphorylate it, it becomes inactive. In this case, right? That doesn't mean that all phosphorylations are inhibitory. In this case, if you phosphorylate glycogen synthase in a particular location, actually to be exact, that outcome is inhibitory. All right. So the enzyme that puts the phosphate on is a protein kinase, and so that's a writer. So it writes the modification. Okay, if you want to remove, this is a covalent modification. So the only way to remove it is actually for another enzyme, a phosphatase, to go on and remove it, the eraser. Right? So in this case, glycogen synthase, if it is phosphorylated, as I said, is inactive. So if you give it glucose, it will not put it into glycogen. First, the eraser has to come in, remove this phosphorylation, which converts now the glycogen synthase to an active enzyme, and now it can make glycogen. Right? So what insulin does to promote glycogen synthesis is to activate the eraser of the inhibitory modification. Okay, so insulin here triggers protein phosphatase 1. Protein phosphatase 1 will remove this inhibitory phosphate from glycogen synthase, and glycogen synthase will go on and make glycogen. The A and B here, forms of glycogen synthase, these are all terms to refer to the inactive and active form, as you see here. A is the active, B is the inactive. All right, so I also told you when we start talking about this that insulin is not satisfied by just, triggering glycogen synthesis. It wants to also inhibit glycogen degradation. It really wants the cell to just put all the glucose it got, just put it away in storage. Okay? So how does it do that? Still, through protein phosphatase 1, it removes another phosphorylation. But here, the phosphorylation, okay, on glycogen phosphorylase, which is the enzyme that catalyzes the phosphorylysis of glycogen and the breakdown of glycogen degradation, when this enzyme is phosphorylated, is actually active. If it is not phosphorylated, it is inactive. Excuse me. So what you really see here are the different effects of phosphorylation. We just saw in glycogen synthase, if that enzyme is phosphorylated, it is off. Here, if the glycogen degradation enzyme is phosphorylated, that is what turns the enzyme on. So by removing this phosphorylation, okay, protein phosphatase erases the activation of this enzyme and converts it to an inactive form, right? So who puts it on? So the writer enzyme of this phosphorylation on glycogen phosphorylase is called phosphorylase kinase. It's another protein kinase that goes on and puts this phosphate on, right? And the kinase itself, okay, the writer itself is actually also regulated by phosphorylation. So we have these cascades of phosphorylation events, right? And for that writer, the kinase to be active, for the activator of the glycogen degradation enzyme to be active, that itself also has to be phosphorylated. And if you regulate this phosphorylation, you can regulate its activity. And of course, protein phosphatase hits both birds, with one stone. It removes these activating phosphorylations on enzymes that eventually trigger glycogen degradation. So it erases all these activating phosphorylations. So you have off the regulator and off the actual enzyme that does the degradation, right? So this is a very elegant way that insulin, through the activity of a protein phosphatase that removes phosphates of proteins, can really block in 2 steps, okay, in 2 different ways, glycogen degradation. And if we see this all together now, we have protein phosphatase, okay, which is again is the eraser of phosphorylations. It's really it's one enzyme with multiple outputs, right? And that enzyme, of course, is the master regulator of that enzyme is insulin. So insulin is able to do this with a single hit here. It affects all these outcomes. Okay, and if you see the pattern here is that phosphorylations of enzymes involved in glycogen degradation tend to be activating. Okay, you phosphorylate the phosphorylase kinase or glycogen phosphorylase, and that activates this enzyme. but phosphorylation, so protein modification of enzymes involved in glycogen synthesis, such as glycogen synthase, are inhibitory. So the outcomes of phosphorylations are completely the opposite in the biological roles here. So this is how insulin works. I know it appears complicated, but just stare at this diagram and things will make sense. So this is how insulin does it, okay? Again, this is how, the one thing that you need to remember about insulin, it's really a homeostatic mechanism. It's a hormone that tells the cell, stay put, okay? Don't change anything. Just put whatever glucose you get, just put it away and leave it for some other time, right? And it does so, again, by promoting glycogen synthesis and actively stopping glycogen from being degraded. So all the glucose that comes in, they just put it away. All right, let's look at another hormone, actually a couple of them, epinephrine or glucagon, and eventually they will lead to the exactly opposite effect, which is to degrade, to trigger, to mobilize your glucose reserves. So these hormones will tell the cells and the organs, okay, move on, burn glucose as fast as you can. Insulin have completely opposite. Go to bed, go sleep. I mean, just put it away. There is nothing to do here. So homeostasis, that's what it is. You maintain whatever state you were. Epinephrine and glucagon will trigger the organism and the cell to be in a different state. Okay, and for that they need to mobilize their energy resources. And let's see how epinephrine and glucagon do it. So they hit an enzyme, adenyl cyclase, which stays inactive, but in the presence of these hormones, it gets activated on the plasma membrane, actually on the surface. And what adenyl cyclase does is to take ATP, but doesn't break it down for energy purposes. It will hydrolyze it, and the AMP will cyclize to give you cyclic AMP, which we have never seen before. And it's really the alpha phosphate. It's in the cyclical form here. And this is what CAMP looks like. It has nothing to do with a metabolic role. It's really a second messenger. It's a signaling molecule, very prominent one. So once this molecule is made, then it will activate another kinase called protein kinase A. Okay, so protein kinase A is inactive, is off usually, but if CAMP is around, it turns on. So you can see how you go from one green to the next green, and then to the next one. So what does protein kinase A do? It activates phosphorylase kinase. Now we're hitting, okay, an enzyme that eventually will lead to activation of whatever it is, phosphorylase, that degrades glycogen. So pKa, so we have this kinase cascades, right, which are very common in signal transduction in cells. So one kinase activates the other kinase, and what that does, okay, so pKa is a writer. Okay? It puts a modification onto phosphorylase kinase, it phosphorylates it, and that turns it on. And once that's turned on, that's also a writer. So we have one writer modifying another writer, and finally, the downstream substrate, which is glycogen phosphorylase, that is phosphorylated, that's active, and glycogen is degraded. Okay? So this is the way that pKa triggers glycogen degradation. And again, by the same target, phosphorylase kinase here is regulated in opposite ways by signals that tell the cell go break down glucose, and signals that tell the cells like insulin, put it away. So, because again, remember that insulin, once through protein phosphatase, wants to remove this activating phosphorylations, and insulin works through an eraser, and pKa works through a writer. So eventually, if you have epinephrine and glucagon, phosphorylase will be activated, and glycogen will be broken down. And through phosphorylases, you're going to get glucose 1-phosphate. And the overall outcome now is this, that whatever insulin does, glucagon and epinephrine do it exactly in the opposite way. So you may ask, who wins here, right? But before we answer this, let's ask the most obvious question, which was the most confusing to me when I was learning this material many, many years ago for the first time. It's just difficult to understand. I mean, simple-mindedly again, why don't we just have one switch to do one thing and that's it? I mean, you go into a room, you turn the lights on, the lights are on. I mean, you want to turn them off, just flip them off. Why do you have switch upon switch upon switch upon switch? The obvious answer is that this is for something that the cells care so much, they really want to control it in detail. And they kind of fine-tune and adjust precisely how much glucose they need to break down or how much glucose they need to put away. Also, in the case of the glucagon, there is something else you need to notice here, and epinephrine, the things that really move the cell from one state to another. It tells the cell to go, don't get out of the resting phase and go on and break down glucose. Every step you do this, there is a signal amplification. So notice all the green things, that you go from one green thing to the next green thing to the next green thing and so on and so forth. So this is very typical of a signal amplification cascade that really flips the switch massively in one direction. And why would you want to do that? Well, what does when do we get epinephrine, running in our system? When you really want to, you're sitting around and you want to get up and start running, imagine some ancient, in old times, you do that when you are chased by an animal or you want to chase an animal. So it's really, you want to go you want to exercise control, which is again different from homeostasis, which is what insulin does. And in this case, with epinephrine, you have a series of activating steps, one activation after another, after another, after another, and then eventually you balloon, the signal is enormous and you can get things done. That flips you from one state to the other. Insulin's job is different, right? I mean, it's to just keep rested, no need to do anything, stay put. That's a homeostatic mechanism. And here you actually see more inhibitory steps that are being put in place. And again, that's a very common pattern when you have a series of activating steps that usually that changes the cell or the organism from one state to the next. When you involve many inhibitors, now here you also have usually a homeostatic mechanism. All right, so that's all I had to say about the fundamental regulation. Now, as I said, in addition to diabetes, which is a massive health problem, obviously, there are many human diseases associated with glycogen storage and mobilization. And you can see a few of them, and they come in many flavors. And you can see various enzymes we have seen, glycogen phosphorylase, for example, transporters, glycogen synthase, and some of the or the debranching enzyme. And this is a summary. It tells you how common they are. Here in 100,000, around 40,000 births, and so on and so forth. And the symptoms are very similar in some ways. Usually you have myopathy, you cannot exercise. So if you cannot mobilize your glucose reserves, obviously you are in trouble. There is very little that can be done. That's the problem. Usually people are advised to change their diet. Instead of having a big bolus of glucose, you have slow release starchy things. Starch tends to be really slower than glycogen. Several, but smaller in quantity meals, and with slow release properties. So at this point, we will stop. I mean, and that concludes the series of videos on glycogen synthesis and degradation.